This image is what we've got to start with, right? Because, let's be honest, Jurassic Park is what always comes to mind when we hear about bringing extinct species back from the dead. This, of course, represents a tale of researchers setting out on a scientific and capital-driven dream to bring back the most iconic species that ever walked the Earth. And it also represents a tale of it all going terribly wrong. Because as Jeff Goldblum's character warns us in the film, when we humans act too closely on our own hubris, we try to control life, life finds a way to break free of that grip. But I'm here to tell you, don't worry, in real life, dinosaur de-extinction is out of the picture. We're not going to be able to work on it. Although scientists have tried to salvage ancient DNA from fossilized remains or even inside the amber-encased insects that have sucked on dinosaur blood, as we see in the film, it's never been able to be read out afterwards because they've been gone for nearly 66 million years. This is far too long to be able to then create a genomic map of what's inside that DNA sequence. It's been exposed to the elements, all these fossilized remains, for all of that time. And after an organism dies, when the cells go into that state of death, a natural process sets in that sets the pH off balance, and then that makes enzymes run around inside the cell and start cutting up the DNA and chopping it into little bits until eventually it's like a bunch of confetti and really hard to string back together. Not only that, it's contaminated because there's bacteria and fungi and all other organisms around it. And so you can imagine after all that time, there's just nothing you can read. The oldest organism that scientists have been able to sequence some ancient DNA from was a 700,000-year-old horse. And when you consider that 700,000 years is just a tiny fraction of 66 million years that the dinosaurs have been missing for, we start to understand why that will remain only in Hollywood and not in the real world of de-extinction today. In our real world of de-extinction, it looks a lot more like this than dinosaurs. So this is, in fact, a picture of the only de-extincted animal to have been quote-unquote resurrected after the last individual of the species died. And this is a clone of Celia, who was the last bucardo, also known as the Pyrenean ibex, which was a type of mountain goat that lived in the Spanish Pyrenees. And although this did happen in the early 2000s, it's debatable as to whether or not it was really a success because this little clone only lived for about 10 minutes. So Celia was the last of her kind because humans hunted this species out of the wild, and she was cared for in a conservation program that had a radio collar around her neck that monitored her every move. And one day it sent out a signal that told her caretakers that something was amiss. So they rushed out into the field to see what was going on, only to find that Celia had been crushed to death by a branch that fell from a tree. So terribly unfortunate way to lose the last member of this species, right? But they had the foresight before she died to have preserved some of her cells. They scraped them off of her ear and from her flank, and they froze them on liquid nitrogen so that that process I told you about where the pH changes and then the enzymes cut up the DNA would not happen. It was perfectly preserved, which meant that 2 years after her death, they could attempt to do something pretty unthinkable, which was to clone her back to life. And so they did this using the same technique that gave us the world's most famous adult animal clone, Dolly the sheep, created in Scotland in 1996. And the process is called somatic cell nuclear transfer. And it involves taking a cell from the animal that you want to clone and removing the package that contains most of its DNA inside that cell called the nucleus. And in Dolly's case, that cell came from a sheep's udder, okay? And because of the udder's approximate relation to breasts, we learn why Dolly the sheep was actually named after a famous country singer with famous udders. This is not a joke. So anyway, with Dolly the sheep, they remove this nucleus from this udder cell, and they insert it into an egg cell from another sheep that's had its native nucleus removed. And then they stimulate it to start dividing with an electric shock, and they insert it into a uterus of a surrogate mom where, if all goes well, it grows into a clone. But science is messy, and the process is a lot stranger and full of failure than the headlines will have us know. So with Dolly the sheep, it took 277 attempts with this method to produce 29 embryos that were implanted into 13 surrogate moms to produce the one famous clone that we all know and love.